Hi everyone, thank you very much for the invitation to join this forum. I think it's a fantastic initiative. My apologies that I can't be there in live, but hopefully you'll find some of our research interesting. So as Julie would have mentioned, I'm Louise Bicknell from the University of Otago, and my lab is, we're gene hunters, so we're really interested in understanding how genetics underlies some conditions, particularly conditions of brain size and height. And so today, some of the research I'm going to tell you about is where some of our really exciting recent stuff, but also where we've had really interesting patients and interesting mutations that have really informed the biology and our thinking about how these proteins might be acting in brain development. So why is brain development and studying brain size so important? Of course, I don't need to convince this audience, but you know, if our brain's too small, that can really impair cognition and cause intellectual disability and impair this intellectual and motor development. And in fact, microcephaly, or a small brain, is seen in many developmental syndromes, often as a bit of a secondary feature, but we're particularly interested in children where that's their predominant feature of their phenotype. Our brains can also kind of grow too big, and this has been associated with autism spectrum disorder. So what we hope is that by understanding some of these more rare forms of brain size, that we can kind of uncover biology and cellular pathways that there might be able to be implicated perhaps in more common forms of brain developmental issues such as autism. And of course, if we ever want to be able to regrow our brains after disease or injury, we need to understand what processes are important in that brain growth and controlling that. And so lots of different reasons to look into it. So the way I kind of got into all of this was during, during my postdoc when I was studying primordial dwarfism. So this is a really extreme phenotype and reduction in size. So this silhouette is me posing as an age-matched control a few years ago with a lady with primordial dwarfism. So this is, as you can see, it's a really extreme reduction. And so when we think about size, both our height and our brain size, it kind of follows a bit of a normal curve, in that you have some a bit taller or heads a bit bigger and some a bit smaller, where this kind of -2 is often viewed as kind of the lower limit of a normal head size. Whereas in contrast, primordial dwarfism is a really extreme reduction in both body size and brain size. So as part of it, primordial is really quite an umbrella term for a number of different syndromes. And so the syndrome that my lab focuses on is called Meier-Gorlin syndrome. So obviously they're small, that's why they're part of that primordial dwarfism group with a short stature. And they often have proportionate microcephaly, though not always. They also have quite uniquely really small ears or microtia, and that be able to see in some of these photos, so in these boys or in this lady here, for example. And they also have small absent kneecaps. So the small ears and small kneecaps is a bit bizarre, but together with this reduced height, that's a really strong clinical diagnosis of Meier-Gorlin syndrome. So we've done lots of sequencing and studying of patients over the years, and we've discovered that Meier-Gorlin syndrome really is a disorder of DNA replication. So the very first gene we found is called, with mutations in Meier-Gorlin syndrome, is called AWK1. And so AWK1 is a protein that's part of a bigger complex of 6 subunits. And this complex binds to your DNA, to your origins of replication. So these are the very first proteins to bind to your DNA all throughout your genome to really kickstart replication. So this all happens kind of late mitosis and G1 phase of the cell cycle. So this all complex then recruits additional factors, CD6, CD1, and then the MCM helicase. And together they're called the pre-replication complex. So these are really, obviously really essential proteins, and they're there to decide when all these origins bind, to lay out that kind of footprint, so that in S phase, these from these sites is when replication will initiate. So there's lots of these that bind throughout the genome. And so following our first discovery in ORC1. We obviously sequenced through all the other members of this part of the pathway, and in fact found lots of mutations that underlie Meier-Gorlin syndrome. So multiple members of the ORC complex, as well as CDT1 and CDC6. So all of those, they had biallelic variants that would cause Meier-Gorlin syndrome. So just when I started my own lab, when I moved back to New Zealand, so this is in 2015, we were lucky enough to get a Marsden Fund grant to try and work out what was going on in that remaining patients. So Meier-Gorlin syndrome is really rare, so we know of about 80 patients worldwide. And we'd found mutations in lots of them, either in this pre-replication complex that I just talked about, or in the next step of denucleation with the pre-initiation complex, where we found one gene in particular, CDC45, that was really strongly linked to Meier-Gorlin syndrome, though with a slightly different phenotype. Those patients often had a funny shaped head, which is called craniosynostosis, where the skull plates infuse prematurely, forcing your brain to kind of push out and push your skull in different directions. But there's still quite a large cohort that we couldn't explain. And so we used 10x whole genome sequencing, so linked read genome sequencing, to investigate these patients. And the reason we chose that particular type of sequencing is that generally it's recessive mutations that underlie Meier-Gorlin syndrome, but we don't always have the parents. So with linked read sequencing, we could separate out the chromosomes, so we could phase the variants, and that allowed us to more efficiently filter variants of interest. And so one finding that's not quite so exciting at a genetic level, but intriguing in other ways, was that we found variants in both MCM3 and MCM7. So remember, I talked about the MCM helicase as being part of this pre-replication complex, so it's not really surprising that we'd have variants here. But these two patients both have mutations in MCM7. So those mutations are, they're both compound heterozygous for a truncated mutation on one allele and then a missense variant on the other allele. So, and yet they look really, really different. So this lady here has Meier-Gorlin syndrome, quite clinically typical in her face, and she has other features like absent breast development that's also commonly seen in Meier-Gorlin syndrome. And the missense variant in this patient lies in the more N-terminal portion of MCM7, and that's required to help bind to the chromatin during this pre-replication complex formation. So it fits in really nicely. That would be a Meier-Gorlin syndrome type mutation. In contrast, this other boy, he has obviously quite a different phenotype. He's got this progeria at birth, so he looks much older than he should. And then he goes on to develop lipodystrophy, where he doesn't have much body fat, as well as adrenal insufficiency. So quite different features to Meier-Gorlin syndrome. But in fact, those features have been linked to DNA replication before, and they're seen in kind of later stages of getting replication going. So for example, in some of the subunits of the polymerases. So while not a Meier-Gorlin feature, it still links in really closely to DNA replication. And what's really interesting is that his missense mutation lies much more C-terminal in MCM7, and that this region of the protein is more linked to helping MCM7 as part of the helicase when it's activated to unwind that DNA and progress the replication fork along the DNA. So it's a different stage of replication to where we normally see Meier-Gorlin syndrome, and that kind of, so the mutation and the phenotype lines up really nicely with a different function. So it's just really interesting, a great example, I think, of how really similar mutation profiles, but just slight differences in where they are in the protein can cause really different phenotypes. The other really intriguing and slightly frustrating at times finding that we had was in a gene called DONSON that we have a bit of a love-hate relationship with, because I've studied it for so long without really realising. So we found 4 patients from our cohort, again with biallelic variants, and they generally seem to be missense variants, so changing amino acids, either homozygous or compound heterozygous fashion. So the reason I have a bit of a love-hate relationship with DONSON is that I've actually studied it for a number of years in a slightly different context. So just a couple of years previous to these Meier-Gorlin syndrome findings, we published mutations in DONSON as the cause of quite a strong cohort, a large cohort with a really severe microcephalic phenotype. And this is in collaboration with Grant Stewart and others. So these are some of the photos of the microcephaly patients with mutations in DONSON. So here's their MRI scans with age-matched controls along the bottom, so you can really appreciate how much smaller their brain is, despite it actually looking pretty normal otherwise. It's just smaller. There's no gross structural abnormalities occurring in these patients. Facially, they look pretty normal actually, though some have quite a large nose. This can sometimes just, the nose can be made to look bigger if you've got a particularly small head, say like in this patient, so it's a bit hard to know whether that's something of particular interest or just a kind of a consequence of that small head. And here's some of their growth parameters. So we can see that when they're born, their head is already smaller, so this delay in growth has started really early in development in utero. And actually they are a bit shorter or smaller than other babies. This dashed line here is -2, so the lower kind of limit of normal. But not extremely so, and certainly not in proportion like we'd see normally in Meier-Gorlin syndrome. As they grow, this brain size has continued to get worse. There's a really progressive microcephaly and it just can't keep up with the rate of growth you'd normally see. But again, their height, while definitely smaller, it's not so slow. So they have this really extreme microcephaly with a mild reduction in height. And so what does DONSON do? So when we initially found all these microcephaly patients, there wasn't much in the literature on what DONSON did. I think there's only about 3 papers or so. But through this fantastic collaboration with Grant Stewart from Birmingham, we were able to show that, that DONSON lies at the replosome. So DONSON actually binds to that MCM helicase and acts as a bit of a surveillance protein. So if there's any issues with the replication fork stalling or slowing because of lesions, then DONSON signals to ATR, which is a really important kinase in the DNA damage response, which then phosphorylates and activates lots of other proteins to kind of get the cell to go stop and to make sure that any lesions get fixed. So it's a really essential protein, but it is acting a bit later than we'd normally see for Meier-Gorlin syndrome, right? Where Meier-Gorlin syndrome seems to be more about organising where replication is going to start and getting that all organised rather than helping that progression of the replication forks. So when we look at the variants that we found in our Meier-Gorlin syndrome patients, we do see that they tend to lie more N-terminal in the protein, apart from one right at the very C-terminal, of course, but generally they seem to be more N-terminal, and so we wonder if that's got something to do with why we see either these 2 different kind of phenotypes appearing of either microcephaly or Meier-Gorlin syndrome. So the structure of DONSON hasn't been solved, and it doesn't seem to relate to any other protein. There's no obvious domains at all that anyone can find in DONSON. It's really a bit of a unique protein. So I wonder whether there's something a bit different about this part of the protein that could be informing on the different phenotypes. We do see that the variants that we identified, that those amino acids are really strongly conserved throughout evolution, suggesting that changing those could be deleterious. So when we had been working on the microcephaly cohort, we saw that for missense variants, that obviously DONSON is a nuclear protein if it's involved in DNA replication, so most of the time DONSON should be found in the nucleus. And we found from those microcephaly missense variants that if you put those into an overexpression analysis, that you could disturb this. In fact, those missense variants were causing DONSON to remain much more in the cytoplasm, so somehow blocking the ability of it to get into the nucleus. So we tested our Meier-Gorlin syndrome variants with the same assay and saw the same effect. So that a huge reduction in how much DONSON was in the nucleus. There's one particular variant where we had a bit of an unusual result, this arginine 211 cysteine, and that we could see that while DONSON could get into the nucleus, it was forming these quite large aggregates. So again, it's not localizing perhaps where it should versus wild type DONSON. So that could be telling us something more about what that variant, the effect that variant could be having on the protein. And that's something we're going to look into further. So we've shown that these variants definitely are deleterious, and so they're very likely to be pathogenic in these patients. But then what about that phenotype? So we plotted the difference between the ratio between head size and height in these patients. So here are the microcephaly patients, and that ratio is quite high, reflecting that really extreme microcephaly they were showing compared to their more mild reduction in height. In contrast, in our Meier-Gorlin syndrome patients, that ratio is much closer to 1, reflecting that more proportionate reduction in size. And in fact, when we plot this data in a slightly different way, so here in grey are the microcephaly patients versus the green of Meier-Gorlin patients, you can see that one patient in our Meier-Gorlin syndrome cohort has a completely normal head size, despite another patient having some of the smallest heads that we've ever seen in children that are alive. So I think it's absolutely staggering that you can have such a wide spectrum despite quite similar variants in the exact same gene. And that's something that we're starting to look into in more detail, perhaps trying to understand what might be going on there. Because of course everyone can say, oh well, there's just a wide spectrum, but actually the patients with DONSON mutations in our Meier-Gorlin, they really are clinically Meier-Gorlin syndrome. They have the small ears, they have the absent kneecaps that we see in Meier-Gorlin syndrome. And so to us, it's either that, yes, these mutations could be acting differently and impacting DONSON in a different way, or that DONSON could be doing something different in the cell. And maybe it's acting not just at the ribosome, tracking that progression, but what if it has a second function much earlier on, much more linked to the other Meier-Gorlin syndrome proteins that we've seen before. So that's something that we're starting to look into now in the lab. So the other example that I wanted to share with you, which is really, really exciting for us, is, and it starts with just one girl from New Zealand, and this is why a great example of how just an interesting patient and an interesting mutation can lead to a whole new big project. So she had quite mild microcephaly, so her head size was kind of -2 to -3 standard deviations below the norm, so normally not enough to get me that excited about a phenotype. And she also had quite strong intellectual disability. And so we did exome sequencing of her and her parents, and we found de novo missense variant, so a variant not present in her parents, in a Histone H4 gene. So there's 15 Histone H4 genes in the genome, 14 encode identical protein. And so while this is interesting, because histones of course seem a bit interesting, that fact it was a bit kind of unclear to work out how exactly this might be doing it when we would expect so many of the genes to kind of rescue this problem. So I turned to the literature and had seen that just previously, just before I'd found this, that a group from the Netherlands, so Heijs van Haften and Federico Tessadori, had published 3 patients from 2 families where they had a different but still kind of similar-ish missense variant in a different Histone H4 gene. So again, these were de novo variants, and they impacted a really conserved, a critically important residue that's for post-translational modifications, this lysine 91. And their patients had a much more severe phenotype, both in terms of their growth profile and also their dysmorphology. But I got in touch with Heijs and Federico and shared our findings and said, you know, what do you think? And should we try and find out what might be going on in our patient and whether this could be something exciting? So of course we only had the one patient, so we needed more genetic evidence. So we used our clinical networks, gene matcher, this dating site, as I call it, for geneticists, and we trolled through all the different databases around to try and find more patients. And from starting with this one single patient, we now have 28 patients from 9 different countries that all share this common neurodevelopmental disorder. So they all have intellectual disability, developmental delay. Most have this quite mild microcephaly, and some also have things like autism, epilepsy, and ataxia. There are also some non-neurological features as well, but by far the most concordant is this impact on brain development. And what we've found is, in fact, that we can see variants in 5 of the 15 different Histone H4 genes, despite them all encoding identical protein. And what's more, because they encode identical protein, we can kind of lay them on top of each other, as you can see here at the bottom of the screen. and in fact see that actually the variants seem to impact the same spots again and again. For example, we have 6 patients here that all show the exact same de novo variant. This is a really striking finding and really exciting for rare disorders. So not only when we look at the protein level, we never see any variants in this N-terminal tail where all the modifications that people routinely study are found. Ours lie in this core globular domain. And in fact, they cluster into 3 main locations. And those clusters and those locations relate to where this histone H4 makes contacts with other proteins or components. So for example, this arginine 45 is a really, in fact, a previously studied mutation and makes a really critical contact with the DNA as the DNA wraps around the nucleosome. Some of our other clusters either is where the protein would normally interact with histone H3 because H3 and H4 form a dimer within the nucleosome, or where histone H4 interacts with chaperones. So of course, the histones need to be removed, nucleosomes need to be removed from DNA to allow for either DNA replication or transcription. And so chaperones coordinate this process, and different chaperones all kind of bind at similar spots. And where they bind is where we see the clustering of our variants. And in fact, some of our particular variants we see make really important protein contacts with those chaperones, predicting that if you change these, that would be deleterious to functioning. Because we have such a rich clinical cohort, we can look at and see if we see any patterns. So these are some of the lovely faces of these patients, lined up from N-terminal variants through to C-terminal. What we generally seem to notice is that as the mutation is more C-terminal, they seem to have a more severe facial dysmorphology. But as we get new patients in, that seems to just make things a wee bit more difficult to see if there's a clear pattern or not. And so we're still working on this. We can look at all their growth parameters and see that as expected, because of how they were recruited, they all do have a microcephaly, and this seems to get more progressive and worse as they age. But actually their height seems to be rather on the smaller side, pretty normal. We separate them out into whether to see if different genes cause the more severe phenotype, or whether clustering a different position in the protein, is responsible for some of the more severe microcephaly or short stature that we could see, but we couldn't see any obvious patterns, suggesting it seems to be there's no obvious genotype-phenotype correlations at this point. So Federico Utrecht did some zebrafish work where he injected wild-type histone H4 into developing embryos to see how they would affect development. And while injecting wild-type histone H4 is tolerated pretty well by the embryos, injecting histone H4 containing one of our patient mutations isn't, and we do see a significant disruption in development. So that also suggests to us it's not the overall amount of histone H4, because you can inject lots of it of normal histone H4 and be okay. There seems to be this ratio between wild type and mutant H4 that is really the critical factor. And what's really nice is this is confirming up for all the variants we've found so far, this assay, that they are in fact deleterious to development, confirming their pathogenicity in our patients. So why do we see, when there's so many histone H4 genes, why do we think that some are causing this neurodevelopmental condition that we've discovered that others don't? And we think maybe it comes down to expression levels and this ratio idea. So if you've got a really high ratio between wild type and mutant H4, so you've got lots of mutant H4 floating around, this really impacts the ability of these nucleosomes to come on off the DNA as required, and that can impact your brain development. In contrast, if it's not so highly expressed, the gene, so you've got less of the mutant H4 floating around, we wonder whether actually these could pose risk alleles for more common conditions such as autism, which we see in some of our patients anyway. And that it's not enough to severely disrupt brain development, and you need additional factors to kind of cross that threshold and cause autism or something similar. And that's something we're starting to look into, both from a cellular perspective, but also using kind of large genetic data sets. And we're lucky to have just been funded another Marsden Grant to look into this in more detail. And so just to conclude, what can we learn from our research? We obviously can't expect to cure reduced brain size or microcephaly. It starts too early on in development. What we're really passionate about is being able to give the family some information, either an answer in the first step, so we know what's causing that condition and the expected risk of having another child with the same condition. But because we're gathering from these larger cohorts, to give them more information about what they can expect. So for example, in that Histone H4 project, the oldest patient we have is in their 50s. So we're able to say to the family of a child who's only 5, for example, we know that you can expect your child to grow to be at least 50 years old. And that's really valuable information for that family to have. We're uncovering some perhaps, I wouldn't say new biology. Histone and chromatin's really strongly linked. It's always been linked to brain development, but I think it's really striking about how everyone's always focused on these chromatin remodelers and their role in brain development, and no one's thought about the histones before. And here we've found through our genetics that actually the fidelity of those proteins is still really, really important and shouldn't be ignored. And through that, we're going to do lots of, in our new project through our Marsden, lots of ChIP-seq and ATEC-seq to try and understand how those variants are impacting transcription and DNA replication and chromatin accessibility in neural stem cells. And with that, I'd just like to thank everyone. Here's a picture of my amazing lab this year. I just sit in the office all day, whereas they're the ones doing all the hard work. And we've got some fantastic collaborators around the world, but by far the most important thanks is always to the clinicians, the patients and their families who, without their DNA and their goodwill, we wouldn't be able to do this project. I'd also like to thank all of our funding bodies. And again, thank you very much for listening. And if you've got any questions or comments or feedback, please do get in touch either through email or Twitter. Thank you very much.